From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on September 19th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 109th episode of Matters Microbial. Many thanks to every listener, viewer, guest, and supporter. Also, thank you for comments and emails. My Bio 350 Micronauts at the University of Puget Sound are doing wonderfully. Micronaut Claire has come to terms with her water bottle buddies, as seen here. September 17th was International Microorganism Day, and I offered extra credit to Micronauts to celebrate that event, and they did not disappoint. Many times students tell me that they lack creativity, and I have never found that to be so. Here is an IMD submission relating to recent possible life biosignatures found on Mars. Here is a wonderful IMD submission and haiku celebrating the beneficial microbes found in the human gut. Naturally, this IMD submission represents ice nucleation bacteria as ILSA from Frozen. And finally, this IMD submission points out that when you adopt a pet, you adopt their microbiome as well. It really brings me microbial joy to see these sorts of creations, and I hope that you enjoy them too. I'll put links to International Microorganism Day in the show notes. It's really worth your time. In lab, my Micronauts are honing their observation, diluting, and plating skills using colorful microbes, E. coli containing plasmids encoding pigment genes, as discussed recently on this podcast. Because of their color, we call them forbidden Skittles. Here is a lovely movie showing that GFP is among the colony pigment types. Several of my Micronauts have found strange hybrid colonies, seen here, that are tantalizing to investigate. What are they from? Two cells that landed close together and appear to be the same colony, or is sectoring taking place? Observation is key. The entire exercise resulted in this lovely meme about forbidden Skittles. Do not taste the rainbow. Much more seriously, infectious diseases are much in the news today, including the threat of the viral neurotropic disease rabies. I had discussed the role of rabies in Europe in the 19th century with my Micronauts in class last week and Pasteur's successful attempts at a vaccine. But even today, rabies remains an issue, as seen in this headline from 2 days ago on CNN. So I am pleased to introduce Dr. Rodney Rohde of Texas State University. Dr. Rohde wears many hats, and one of them is the study of rabies. Welcome to the Quality Quorum today, Rodney. It was wonderful to meet you at ASM Microbe and have you asked to join the podcast.
Thank you, Dr. Martin. It was an absolute joy to meet you at ASM. What a great conference to meet so many different Micronauts, as you say. So I'm excited to be on your show today, and I look forward to talking to you about some of my background and my work with rabies.
It's really interesting because you have so many things that you do. And as always, I put a lot of links in the show notes. And when you look at your website and your various web presence, what don't you do becomes the real question. And so I hope people take the time to look at that. By the way, for listeners and viewers, ASM Microbe is the big, ginormous meeting of microbiologists that happens once a year. Are we in Washington, D.C. this coming June?
We are. So Los Angeles this past year, and we're headed to the East Coast to Washington, D.C. for 2026.
Wow. Just amazing. But in any event, even among those thousands and thousands of microbial enthusiasts, I have so many friends and colleagues. And so it's a lovely chance to come together and chat.
Absolutely. Absolutely. Looking forward to it.
Rodney, I think the right thing to do to start with this is to talk a little bit, if you wouldn't mind, about what you particularly do at your institution, just so folks get some contouring of where we're going.
Yeah, I appreciate that opportunity. So just a little dive into my background. Once I finished a bachelor's degree in microbiology and a master's degree in virology back in the year of 1992, I immediately went to work for the Texas Department of Health in Austin, Texas. And this is where I was initiated into the world of virus isolation. I ended up working in rabies arbovirology and in other vector-borne diseases like West Nile virus and things like that. Spent a decade there. Was really one of the greatest times of my life. I got to spend a few times at CDC as a visiting scientist. Was part of the inaugural World Rabies vaccine program that we'll talk about shortly. But after about a decade there, Mark, I had been teaching on the side as an adjunct teacher at Austin Community College in Austin, teaching introductory microbiology to health science majors and biology courses. And never in my life, Mark, did I think I'd be Professor Rohde. I hadn't planned for it. I was loving working where I was working at the Department of Health. CDC was like, the Taj Mahal of everything I'd ever thought about doing. But I just kept finding this love for teaching and mentoring, and it kept kind of pulling at me. And in 2001, I had a chance to come back to my alma mater and do a actual microbiology course for a former professor because he was on sabbatical. And while there, this was right during 9/11, by the way, which is kind of seared in my brain because I was dealing with that semester when that was happening. He came to me and said, hey, there is an actual position in the medical laboratory science program, and they're really looking for a specialist in microbiology, molecular diagnostics, and that was my wheelhouse. All of my credentials from ASCP were in those areas. And so I kind of tossed my hat in the ring, never thinking that I would have to make a decision, thinking they're not going to come after me. And lo and behold, as I always tell my students, be careful what you apply for. And be prepared to be offered that position, because that's what happened. And I ended up here at Texas State University in 2002 as a assistant professor on tenure track. And over the last 4 years, I've moved through the ranks, and I'm currently the Regents full professor and University Distinguished Chair of the Medical Laboratory Science program in the College of Health Professions at my alma mater. I'm a proud Texas State Bobcat. And through that time, as you know, that we've kind of talked about, I'm starting my 30th year as an adjunct professor at Austin Community College. So I do wear a lot of hats. I've been a research dean in our college prior to stepping out of that role, and I just love to stay busy. And so my students and my colleagues often ask, how do you juggle all this? And I know you feel the same way. It's just a matter of organization and what I'm passionate about. So Mark, if I didn't teach, then I wouldn't have the passion and kind of the fuel for what I do, both in research and as an administrator, and all the hats, as we've both talked about. So that teaching piece, that mentoring piece, is really the passion for what I do professionally.
It's really quite amazing. My wife used to do administration, and now she runs a museum. So I'm very aware of the lots of different things aspect of it. And for her, she finds it energizing. It sounds exhausting to me, but it sounds like you're in her kind of group.
Yeah, the administration piece, it's a necessary thing for any program to run efficiently. But part of the passion of that is that I enjoy the part of program building. We've recently created a medical lab science minor, which I'm really excited about, to pull in pre-med students and STEM students to introduce them to the world of medical laboratory science. So that piece is really enjoyable. Like most administrators, the things like meetings and hiring and personnel, they're all necessary things to do, but that part of it can wear people down a little bit. But again, that teaching piece, that mentoring piece, and I always tell my wife as well and my colleagues that, the greatest job in the world is being surrounded by young people. and a college and a university
Yeah.
where we have so much diversity and so many great students from around the world that I'm learning every single day when I, come to the classroom or helping a student with advising or something like that.
So my friends who aren't educators will say things to me. Isn't it boring to just teach the same stuff year after year? And number one, it's not the same stuff because my
Right.
field always changes. But number two, as you say, it's this cross-pollination, and I'm not being, all sophisticated when I say it. It really is true that I learn as much from my students when they encounter information than I knew to start with, because my big goal is to get them to understand there aren't any dumb questions. There are questions that aren't thoughtful, but that's not dumb.
Right.
And I learn from every time there's a student with a question, other people have it too. So I will miss it very much is my point.
Yeah.
But I want to emphasize for listeners and viewers that the MLS program, and I just want to say we've had lots of academic microbiologists on the program, but this is part of the mix in the microbial sciences, and it's one that's directly relevant. So for many people to look at that as a career option is a wonderful thing, and how delightful that you're in charge of such a program.
Yeah. Thank you. Thank you for pointing that out, because as you know, all of my degrees were in the core sciences of microbiology, virology, and my PhD's in public health, which I picked up later when I came back to Texas State. But medical lab science is actually a degree I didn't know about.
Mm-hmm.
So part of my story is that this actual program, Mark, was right across the street from the microbiology department, which is in the biology department across the street. And I love my degrees, but one of the great things about MLS, medical lab science, is that if you want to practice in a hospital laboratory, you need a credential.
Mm-hmm.
Much like a doctor needs a credential, a nurse needs a license. So we are credentialed professionals. You actually sit for a medical lab science credential from ASCP or other entities, and then you can practice in a hospital laboratory. So many students don't know that, and so it's a good message. I appreciate you pointing it out.
Mm-hmm.
And my kind of funny, ironic thing is I often can tell this story because in many instances, I was a microbiology student and virology master student. I didn't know about this
Mm-hmm.
field, and I might have went this way because my goal was to work in a hospital originally. But, in the big scheme of things, it all worked out because I got to go into public health and just had a wonderful time working in those areas of rabies and virus isolation and the things that we'll talk about today.
Well, and I do want to say I always put links, and incidentally, Rodney, you were wonderful at sending lots of different links to me. Often that doesn't happen. And I wanted to tell you that this is something that listeners and viewers that might be thinking about careers in this area, there's going to be information on that. And thank
Outstanding.
you. Mm-hmm.
Outstanding. In fact, and you may not even know this. So each year in the United States, we produce about 8,000 or so graduates, but we need about 20,000.
Yes, I'm not. Yeah.
So, the great thing I tell parents as well as their children is that when your child graduates with this degree and a credential, 100% employment rates out of my program for 40 years. I mean, you just have to not want to work if you graduate. It's much like nursing and
Mm-hmm.
other things. There's such a healthcare shortage in these areas, and we need people, Mark. We need excellent microbiologists. We need excellent blood bankers. We need excellent hematologists and clinical chemists coming through these programs, as well as regular micro programs that enter into public health, research fields. More than ever, we need experts out there doing this type of work because of the inundation of infectious diseases and the ever-going evolution and mutation of these microbes.
Absolutely. So if you don't mind, I would like you to give kind of I don't know where the expression thumbnail sketch comes from, but we all say it. Maybe it's because you're sketching it on your thumbnail. I'm not sure.
Yeah, maybe so.
But when we hear about rabies, everyone, and like I said, it's been in the news recently, people never pick up a bat, that kind of thing, and how wonderful to because the name, if you ask people their first reaction to the term rabies, the terms and associated with rabid and that kind of thing, it really has a negative reaction. So could you talk a little bit about the disease?
Sure. So Rabies is, as you mentioned earlier, a neurotropic virus. So ultimately, what's interesting, I often tell people it's one of the most fascinating viruses to me, but it's also one of the most diabolical because of how it kind of works through your body in a pathological way. So wherever you get bitten, whether it's a human being or another animal, the bite of another warm-blooded animal is what triggers that initiation. So a rabid animal can be any warm-blooded mammal. It can also be a bat, which you mentioned. So in the United States, bat bites, usually unrecognized bat bites, are the most common route of infection. And you're right, most people in the United States are very fortunate because typically we have 1 to 5 deaths annually, nice low number. But when you look globally, Mark, 50 to 60,000 people still die from rabies around the world. And these are mostly in places like Latin America, Russia, Asia. And it's really because they don't require strong domestic animal vaccinations. So when you travel, one of my safe comments to people sometimes is when you're going to places outside of the U.S., even that cute puppy or that cute feral cat, things that are kind of roaming around, you should really be careful because actually a lot of the cases of rabid humans that come back from traveling do occur from a dog bite, actually. We don't really see that in the U.S. because our dogs and cats and cattle and things like that are mostly vaccinated. But once that bite happens, the virus starts moving throughout the peripheral nervous system. Eventually it gets into the central nervous system and ultimately reaches the brain. And, there's a lot here we could talk about, but what's kind of an interesting facet of rabies is there is something that triggers that virus pathology that it will excrete virus through the saliva. And so most people know about this with respect to, maybe movies like Cujo or in our time, Old Yeller or something like that.
Sure.
And so even though that's the vision from first people, that bite, maybe a vicious bite, in reality in the U.S., it's mostly unknown occurrences when they pick up or don't even realize they put their hand into a brush pile or around a tree. And the bite of a bat is much like incisor hypodermic needles. You really can't feel it sometimes. It doesn't always draw blood, and there's often not a lot of pain because it's such a fine, sharp puncture. And so tragically, many of these are children. They don't think about it. They don't report it. They may not even, their parents may not believe it because some of these cases will be about children telling their parents, Mom, a bird flew into my room last night. And you're like, okay, whatever, right? And so tragically, they may have encountered a bat. And so once you're bitten, and again, these symptoms can vary based on where you're bitten and all sorts of immunology factors, but generally looking at about 8 to 14 days, maybe up to 30 days when symptoms appear. And once symptoms appear, Mark, it's basically mortality. It's really, something that I tell people all the time. Rabies is basically 100% fatal, but it's also 100% preventable with vaccination. And, just to mention here, we're coming up on September 28th, which is World Rabies Day. So great timing to visit your show. And that's a day that, is about awareness, visibility around rabies education. And it's a commemorative event of Louis Pasteur's death. And so it's kind of a homage to his vaccine that has saved millions of people from rabies deaths.
And it's a funny vaccine because they didn't know very much at the time at all.
Yeah.
And when I talk about it in class, and there's no reason to go into the old vaccine, but I had to emphasize to students, this made Pasteur a hero because of the number of stray dogs and the amount of rabies affecting children in Paris primarily, right?
Yes. And what's interesting, I just visited Paris this past May. I had an opportunity to participate in an early study abroad preparation area. And unfortunately, the Pasteur Museum was closed for refurbishing, so I didn't get to visit. So I hope to get back to that. But you're right. In those days, there were a lot of rabid animals, and it was a very scary type of thing. Most movies even today are based on, I Am Legend or, the zombie apocalypse. And they tend to be linked to rabies
Yeah.
because of that diabolical feature. But past year, back when that happened, it was almost out of necessity because a royal was bitten by a feral dog. And I believe his name was Joseph Meister, if my memory is correct. And he basically was not ordered, but basically pressured to try his vaccine on this young boy because it was a royal, and it ended up saving this young man. And so the rest is history. There's been many iterations of the vaccine, and now it's mostly a cell culture derived vaccine. But he was literally using like spines of rabbits,
Yeah.
and, creating this vaccine. Really old school stuff, but it worked. Again, no more shots in the abdomen. That's gone away decades and decades and decades ago. Now it's basically a series of shots in the arm and usually some human rabies immune globulin around the bite.
Yeah.
So sometimes you'll add that around the bite to neutralize any virus in the bite area. But it's much like a tetanus shot. And you usually receive about 4 vaccines, maybe a 5th if you're immunocompromised. So it's like day 0, 7, 14, 21, and then you might get a 5th one if you need it. But it's life-saving.
Yeah.
It's life-saving. So in most instances in the U.S., if there is any doubt from a physician that there's some unknown bat exposure and they can't rule it out. So an important point here, Mark, is that if this ever happens to you, if you can call animal control or a veterinarian or even the police, because you don't want to handle the bat or any animal that might be suspicious, right? We want to avoid strangely acting wildlife because they might be, that might be a problem. And if they can capture the animal and test it and it's negative, then you don't need the vaccine series.
Right.
But if you can't capture it and it disappears, most of the time they're gonna lean on the side of caution, and you're probably going to get that rabies vaccine and post-exposure prophylaxis. Because it saves your life.
My understanding is anyone who handles a bat basically should get the series. And
right.
I didn't know what you just mentioned about the bat bites being essentially, I'm not going to call them benign, obviously, but unnoticed. And I think that's a really good thing, good information to put out there. At the same time, I want to say that doesn't mean that we have to be petrified of animals, but
of course not.
I do have a question about bats. Are they asymptomatic for rabies if they're carrying it?
They are. Wow. They are. They don't really show signs. Now, some of them will become, it's a percentage. Mm-hmm. Some of them will become sick, and that's often one of the more dangerous things because they'll fall. And they may act like they're still alive. And so not only for children, for example, or even adults that might want to help, right? A lot of people are very helpful with sick wildlife or sick animals, but that's when you really should take caution.
I agree.
Because it might be nothing, but it could be rabid. And so you want to be really careful with that. And, that mention of that unrecognized bite, there's an image that you can share that I have shared with you about how a researcher actually showed that by taking a bat that was not rabid and actually had that bite occur on a finger, and there's a picture of it that shows it blown up. And what's interesting is there's no way any physician or even you could see it without serious magnification. So, it would be almost impossible for a physician to really recognize a bat bite. It's like the finest little needle prick, and it's really difficult to see. And so that image has the skull that shows kind of the canids, the teeth of a bat, and then it has a hand with a finger there to kind of show you this really important educational piece around why you should not handle bats.
I'm also really, when I hear about a small number of cases per year, and many people will hear that and they'll think it's not an issue, forgetting what it took to get to that.
That's right.
And your information that you gave me, and I'm going to look up some information and put it in the show notes about the prevalence of rabies and rabies fatalities worldwide, really says something. And I want to thank you for that information.
Absolutely. I really appreciate the opportunity to educate your listeners and others. It's so important.
The question that occurs to most people about any type of virus when it impacts behavior, and especially if it's swift and almost always fatal, is what's in it for the virus, with the understanding that viruses are not sentient. At least that's what I say here. So what's in it for rabies to have such an effect on the host?
Yeah, what a great question. It's kind of a different way of thinking about it, almost from the side of the virus. And I mean, ultimately, I always tell people microbes exist to exist, right? Their ultimate goal is to, I mean, a typical pathogen, especially, their goal is to infect a host, amplify, make more virions, make more virus or more bacteria, whatever it is, and then jump to the next host. And that's their goal, to survive, to reproduce, to survive. They really have no other goal. And unfortunately, some viruses and bacteria and fungus and others harm you or, sometimes kill you. And so the idea is that really a virus wouldn't want to have a dead end host. That's not in its best interest. And so by having bats and other animals that can kind of carry that virus and shed it for long periods of times and perhaps not die is a way to keep the virus alive or viable in nature. And this is also an explanation I tell people about expectations to eliminate SARS-CoV-2 or anything else, especially RNA viruses. When you have animal reservoirs that you can't eliminate, it would be near impossible to eliminate rabies from bat populations. They're mobile, they fly. It's just almost impossible to think of ways to get rid of it. You are never going to probably eliminate rabies. Unlike the contrast to something like smallpox, which was only a human host, if you create a vaccine that eliminates smallpox in human beings, you can eliminate the virus. Measles would be another one that we could potentially eliminate if we could get on board with vaccines and keeping those current, right? We could literally eliminate measles. Polio, we've almost eradicated polio, but we have to stay, very stringent and sustain that effort because that virus, Mark, it's waiting. It's waiting somewhere in wildlife. It's waiting somewhere in other insect host or the water if it's bacteria. So we have to be very vigilant in doing that. But ultimately, the diabolical nature of rabies is to have that host become aggressive in nature. Some of the chapters I've written, some of the papers I've written, if you're interested, kind of talk about this, that it invokes an aggressive behavior that makes that kind of biting effect so that the virus jumps from one host to the other.
So you have ways of testing for rabies, and what are those ways?
Yeah, so this is something I often tell my students I never dreamed that I would be doing. I certainly was never trained or educated in this with my first 2 degrees, but when I went to work for the Department of Health, this was the area I ended up in. I first worked in virus isolation, which was cell culture based. It was all the human viruses of clinical significance. Learned a ton, Mark, on classical virology, culturing and monoclonal antibody typing. This was right when molecular PCR world was opening up in the early 90s, so it was a great time to be in testing. But I had this opportunity in 94 to move into rabies, arbovirology. It was with Zoonosis Control Division. And the way this works is that for an animal, most of the time, so I'm talking about animal testing here primarily, is you really do need brain material. That's where the virus ends up at. That's the most prominent area. It's really the gold standard to do something called a fluorescent antibody test. And if you could imagine a glass slide, and one of the things I learned, which I never thought I'd do, was animal necropsy. So I'd get geared up, suited up. It's a level 3, level 4 virus, and we'd work under these massive hoods. And I learned how to open up animal skulls, from dogs to skunks to bobcats to horses. We even had a sad instance of a Bengal tiger one time from a zoo that had bitten somebody. A really sad situation because a human stuck their hand in and got bitten, and they had to put the animal down. Terrible. But just all sorts of animals, goats, cows. I mean, anything you can think that's a mammal. And some days in the summer, Mark, in Texas, we'd have 100 heads to open because there was a lot of animal. If you had an exposure, you had to rule it out. So the veterinarian or someone would send it in and we'd test that. So once you had the brain, and you have some slides to look at this, we would open that head up, we would get the brain out, and really the primary area of the brain you want to test is the brain stem. But sometimes you would also test the cerebellum and a little bit of the hippocampus. And so you'd put those pieces on the slide, you would take it in, you would fix them with acetone, and then you would add a mixture of rabies antibodies. And as most people know, if there's a specific match, that antibody, right, in the shape of a Y, which would have a FITC fluorescent compound on it, fluorescent, and it would stick. And so if it was a rabbit antigen brain material, that antibody would stick to it, and you would rinse it, incubate it, rinse it, and then you would take it to the darkroom, and with ultraviolet fluorescence, you would see this bright green starry sky, apple green fluorescent, and that was a positive rabies sample. If it was not fluorescing, then it was negative. We also did monoclonal antibody panels. This gets into more of my expertise where we typed the rabies variant. So everybody's heard the word variant during the past pandemic. And we were doing this, many years ago where we wanted to know, was it skunk rabies? Was it a particular bat rabies? Was it canine rabies? Because each reservoir of animals sometimes has its own strain or variant of rabies virus. By the way, all those variants still are treatable with a vaccine. It doesn't matter which strain it is. But for epidemiology, it's kind of important to know what's going on. And so we also did that. And then I also had the wonderful time of, this is when I got to travel to the CDC and learn from rabies legends, Dr. Charles Ruprecht, Gene Smith. These are people that have been working in the U.S. rabies world at CDC for decades. Great mentors. I still work with them and publish with them, and learned how to start up this new, wonderful platform of PCR-based testing. I did Restriction Digest to distinguish between coyote, fox rabies, kind of between those, as well as bats and skunks. This became really critical during our 2 rabies epizootics from fox and coyote rabies in Texas, which we can talk about if you like. But that was, that was kind of the daily grind, was opening animal heads, making slides, sitting in the darkroom for hours, and then maybe typing them. The typing part was really focused on what I did later. And then on the other side of that, a lot of what I did with the other guys, had some great colleagues in this area, by the way. We did a lot of the mosquito testing for St. Louis encephalitis, Western equine, Eastern equine, just all of the mosquito-borne viruses were kind of in that zoonosis area. I had so much fun working in this area. I often miss it, but academia pulled me even stronger. But I would go do it again if it was an opportunity to go do that and kind of enjoy working in that area. And sometimes just working in that laboratory is, some of the more fun memories that I have.
It's interesting that you say these things because I have never worked on a microorganism that is associated with health issues. I was essentially trained as an environmental microbiologist, right?
Yeah.
Or a soil microbiologist, more accurately. So the whole idea of working with something that could potentially cause disease has been interesting to me because no one would ever question that work being important because it's all important. I know that.
It is.
But for everyday people, they really seem to connect with that kind of thing, which is why, again, hearing about doing that for a living has been important. I did have a question, though, about the rabies test, how often you get a false negative, I should say a false negative, because as you mentioned, and I want to emphasize to listeners and viewers, the rabies virus essentially hides out in the nervous system where your immune system can't easily see it, which is, I have levels of fascination about that. But so if you take a brain sample, is it possible that the brain sample that you get doesn't have the virus present, so you could have a rabid creature and still come up with a negative?
It's less than 1%. It's very, very rare. That's why the brain the brain and the FA, the fluorescent antibody test, is still kind of the gold standard, Mark.
Mm-hmm.
Although, just to get into the weeds a little bit here, when we would read those slides, there would be times, we had two sets of eyes. You always did two people, kind of as quality control. You had lots of different positive and negative standards. So to get into the weeds a little bit here, you had 1 plus, 2 plus, 3 plus, 4 plus fluorescence.
Mm-hmm.
And you had to train on that with mentoring. And so everybody was kind of seeing the same things. But if there was any doubt, and we did have it occasionally occur where we called things plus minus, or, they're a little strange looking. Interestingly enough, some of the brains that we would get, think about Texas, summertime, it's 100 degrees, animals would be decomposing.
Yeah.
And so sometimes we would have to reject, just like in a hospital, we would reject the sample because they were so decomposed with bacterial decomposition, and bacteria fluoresce. They autofluoresce, so it would give you this false positive. Again, false positives are better than false negatives, as you know. At least with the false positive, you treat somebody, they may not need it, but you're treating them. False negatives would be detrimental in rabies because you would miss the diagnosis and you would not treat, and that person could potentially die. Mm-hmm. So that was you never wanted that to happen. So if there was any type of doubt, we often would put that brain into cell culture, neuroblastoma cells, which are brain cells, and we'd amplify. Back then, Mark, this is kind of less common now, but CDC and some research labs still do this. But we actually had mouse houses. We had an animal room, and for really weak specimens or mosquito viruses that we were trying to amplify, we would inoculate intracerebrally and intraperitoneally these mice with virus suspensions to amplify the virus. And then we would sacrifice the mice and look at that brain material. That was super rare, but that could happen. And then fortunately, when PCR came around, the powerful technique of amplifying even one copy, theoretically, of a virus really helped, kind of calm those fears. But it's, the potential could be to miss that. That would be terrible.
Yes.
But that's why when you work in that world, you take so many efforts to have an option, several panels of types of tests to kind of rule things out. And then if you really can't make a decision, you still could ask that physician to go ahead and treat. It's not going to hurt the person to get vaccinated because it could save their life if you did happen to miss it. And, in reality, Mark, when humans, occasionally we would test human brains or human saliva with PCR testing. Interestingly, you would pluck hairs from the back of the neck. Yeah. Because the rabies virus would get into that root of that hair. Or you might take a skin biopsy or something like that. Pretty rare to take a brain biopsy of a human, but maybe if they were in there doing something else, saliva. But the FA of the brain was still the most accurate test for animal testing. So that's still done. There is some new amazing molecular panel testing out there now that's being looked at for global use that has universal primer sets that would amplify any variant out there. So those are pretty exciting. They're still being looked at. So hopefully one day, all over the world, there will be cheaper, easier to use diagnostics, maybe even in the field. I mean, that's the goal. So countries that are struggling with rabies, we can have cheaper alternative types of testing that they can use that would be helpful to save lives.
So it's important for listeners and viewers to note that this particular neurotropic disease has been studied for a long time. There's a wealth of data about it. There are improvements that are taking place on that data. But we have a protocol for looking at things. And this is very different than a disease that occurs seemingly suddenly. And so what I want to emphasize is there is a great deal to be confident about in the way that we handle rabies in this country. And that's what I want people to know, especially in the current climate. But what I would like to also ask you, and moving into something that I know you've been involved with, is the idea of surveillance and possibly reducing the incidence away from people.
Yeah, so this is probably one of the most amazing programs I've been involved in. I often tell my students I was in the right place at the right time. I had the right skill set, and I always tell them, don't be afraid to step into a role, even if you're, a bit hesitant. I was relatively a brand new worker. I'd been out of grad school, out of my master's program for 2 years. Texas had two big rabies epizootics occurring. An epizootic is an epidemic in animals. And so we had a huge rabies problem in coyotes down in South Texas as you get towards the Mexico border. And we also had a huge problem in Texas gray foxes, which was more in the west central Texas area. And again, I've shared some slides with you to look at these particular outbreaks. So in the late 80s, early 90s, we actually had several deaths in humans due to these types of animal bites.
Mm-hmm.
And so back then, it was Governor Ann Richards. She declared a rabies public health emergency, and that helped open up some funding and some other ways to move forward. And there's so many people to thank on this, so I'm not going to be able to, do justice to the biggest group of them. But it was termed the Oral Rabies Vaccination Program. And this came out of Canada and Europe. Dr. Ruprecht, Gene Smith, my former boss, Dr. Keith Clark, Dr. Susan Nill, just many people. I'm going to miss so many people here, but some real giants in the world of looking at recombinant rabies vaccines. And what this was a piece of the rabies virus, the glycoprotein, the nucleoprotein, was inserted in a vaccinia virus. And this was all field tested, so we don't really have the time to talk about all the animal trials. So they did a ton of prep work to make sure this vaccine was safe, that it was safe to use in animals. And so all that work was done and documented and published. And then Europe started using this for different rabies in animals. And what Canada was doing at the time, they were flying these twin otters, these big yellow airplanes that they used to put out fires with or do other types of counts of animals. So they're very sturdy, reliable planes. And they made these baits. And again, I sent some images of these. They look like dog food baits. They're about the size of, I don't know. It's like a cube shaped, and they were hollow in the center. And in that center, they would put this liquid vaccine that was a recombinant vaccine. And they had all this science behind of how you drop these baits out of an aircraft. So if you can imagine a plane flying like this across the sky, about 500 feet from the ground, so it's pretty low. And they had these conveyor belts kind of going the opposite way towards the tail of the plane. And it would drop baits out of a chute. And it was based on the plane speed. It was based on the elevation. And someone like me or others would sit in the cockpit on the side of the pilot, the co-pilot, and they would operate these devices so that the baits were being dropped at a certain baits per ratio per square mile.
Right.
So it's very scientific. But basically, we were just feeding coyotes or feeding foxes all over Texas. And they started this in 1994. So I got to be a part of this amazing program. And over the years, as more and more coyotes and foxes would be immunized, we created a living animal immune barrier, if you can imagine that. And we started up higher on the tip of Texas. Texas has that kind of crook at the bottom. We started up here, and then we would move it based on case counts. We would push it down to the Gulf of Mexico. And so it took several years to push it down. And we just went every year in January because that's the coolest time of the year. And we would take over small towns. We would fly from dawn till dusk. I forget what the number is now. We have flown around the world multiple times. We have dropped tens of millions of baits. And we have eliminated canine rabies in Texas. But what's interesting is during all of this, back when I was working, what my primary job was, I got to participate in that, which was amazing, Mark, because who would ever think a microbiologist would get to leave the lab and get to go do these things? And so I was really one of the first emerging infectious disease kind of hybrid professionals. That's how I describe it. I got to play in the lab because I needed to know the typing. Was it fox rabies? Was it coyote rabies? Did it not matter? Was it skunk or bat rabies? So we documented that with GIS markings using latitude and longitude, and we would devise our lines of baiting based on those tests. So I got to play in the lab and figure all that out, and then I also got to go fly, and then we also got to work in post-drop surveillance where we would collect samples from animals, both from the teeth, because the bait had a tetracycline biomarker, so you could actually, this is a whole other area, they would do slices of the canines, little slivers, and it would fluoresce. It's like counting rings on a tree, Mark. It would be like green rings. We had coyotes that would eat 20 plus baits. They were hungry. And then we also took serology. We'd take blood samples and we would look for antibody titers. So we've published these data over the past 20 years, and it's hard to believe it's been 20 plus years that we've been doing this. But I often tell people very humbly that it is truly, I think, one of the greatest public health feats, at least in our country, but maybe globally. We have eliminated canine rabies at its source in Texas. There are no more canine rabies. We have bat rabies and we have skunk rabies, but we've eliminated canine rabies. Our ultimate hope would be that other countries would adopt this.
Sure.
So that you could knock down those tens of thousands of cases in places like Latin America and Mexico and Asia and things like that. Because you could save tons of human lives by doing this. It takes a little money. It takes a little willpower. And so hopefully other countries will adopt this going forward.
Well, what I like about it is though there is a technological aspect to it, having to create the recombinant virus and grow it up, certainly the delivery systems are variants of things that almost every nation has access to or can get access to. And this is a wonderful thing to see because there are many types of diseases that have animal reservoirs and you can't simply treat people. I did have a question about rabies vaccine. So the rabies treatment that we have, the one that we're getting shot in the arm, that's not really a vaccine. Well, it is a vaccine, but it isn't a robust one in the sense that it doesn't give protection later, or does it?
You know, it does, but it's something so when we did it, so I've been immunized many years because I work in the laboratory. So any worker that works in a research laboratory with rabies or other viruses typically is going to receive that vaccine to be pre-exposure protected, right? But every year, people that do this are doing titers. So we check that titer annually to make sure you have a protective level. It's 0.5 international units for rabies. And if you're protected, you don't need a booster. If you slip below that, you get a booster shot.
Perfect.
And so some people, it doesn't last long for whatever, everybody's immune system is a little different. I had one person that had to get it almost every year. I went 7-8 years before I really needed a booster. So it just, those are some of those unknowns about the wonders of the human immune system. But some people would have protective titers for 7, 8, 10 years. Others, it was almost like you almost needed it more than once a year. They just couldn't maintain a titer for whatever reason.
When we talk about humans in particular, one of the things that I tell students is when we do animal studies, almost always those animals are highly inbred and they're going to react to things. That's why we're using them. But humans are incredibly diverse, and this is a good example of it. And so the people that required more frequent immunizations, there's nothing wrong with them. It's just that their biology is a little bit different than the next person's. Yeah.
That's right.
And that's the challenge of it. And what a wonderful thing to have been involved with, that kind of treatment.
It's so funny, after having, we've all been dealing with COVID and SARS-CoV-2. It's been a interesting time in history to live through that. But in many ways, what I did 20, 25 years ago prepared me for COVID, because at my university, I became the source as a specialist in virology for questions from the media, from colleagues. I was put in charge of some committees to safely do research, or how do we teach in these kind of concerned areas of aerosolization. So I often would tell people, look, if you have concerns about mRNA vaccine technology or things like that, we've been doing this for decades
Mm-hmm.
in animals, and it's very safe. I mean, who would have thought in 1994 that in Texas we would be dropping recombinant vaccine in the wilderness for animals to eat to immunize them? Nobody really raised any concerns. And so it was kind of interesting to me to see kind of the uproar sometimes around the vaccine technology during COVID, because it's super safe. It's been tested in animals and humans in many cases for decades. And so, not to get too far off
No, no.
tangent here, but it shows how one's background and experience, and I tell students this all the time, just never underestimate your training and your education and your willingness to try to step into a role that maybe you're not totally prepared for. I hadn't been taught anything about rabies testing specifically and what to do, but man, did I learn a ton from experts around the
Mm-hmm.
country and around the world. And it has been the most amazing career path, all because of rabies virus. Has really propelled me into areas that I never thought I'd be on a podcast. I never thought I would host a podcast, which I do now with Contagion Live. Writing, speaking, traveling the world. It's a dream come true for a microbiologist to be doing this. And now really my passion is, as yours is, I think, is accurate science communication to the public, needed more than ever in this community to get the word out about what's safe and accurate and really important for people to understand.
I can't agree more. And, this is something when I would teach introductory biology as well as microbiology and genetics, I would tell students that a lot of times you'll hear people who have no background in an area talk about it, and they don't really know what it is they're talking about. And I'm not being nasty about it. If you were
Nope.
to ask me about the finer points of inheritance law, I don't know much
That's
about
right.
that. I could look
That's right.
something up online. There's a difference that we see today that some folks create a sense of fear, and I appreciate that fear. I do. But stories like the ones we've talked about today, not just the fact that Louis Pasteur came up with what would be an unapproved vaccine today.
That's right. That's right.
And how far it's moved along and how people have worked together to make it happen and evaluated what's taken place. Those are important lessons to keep in
They
mind.
really are.
Yeah. And
They
so
really are. I often will tell students and colleagues and total strangers, I love having these conversations. We need to have these conversations more and more. And I will tell them, when you fly, when you get in a plane across the country or across the world, your expectations is that pilot has gone through rigorous training and education, and you trust that. Why is that any different than a scientist or a physician or an economist or anyone that has expertise? So my goal, and I think it's a lot of our goals around those of us who do this, is that I want people to understand that expertise still matters.
Oh, yeah.
More than ever. Whether it's about vaccines, whether it's about microbiological biofilms from an environmental standpoint, you can appreciate those really important things. Why you need Mark Martin working on these things, right? You need experts understanding nature, animals, the environment. It's really that one health view that we're global, right? Global is local, whether it's rabies or COVID or measles or some other cancer or antibiotic resistant infection. I want the best minds working on that. And when I get treatment for a family member or something like that, I want the best working on that. I don't want it to be guesswork or through fear. So that's my goal. That's the goal as I kind of am getting towards the end of my career. Even when I retire, I think I'll be doing more of this. It's really my passion is to spread the word among anyone and everyone that will kind of think about this and hopefully adopt that position.
Rodney, this has been such a fabulous podcast opportunity. For me, I've learned a lot from today. It has been a joy to chat with you. And again, so nice meeting you at ASM Microbe. And you said, I'd love to be on the podcast, and here you are.
Yes, sir. I thank you so much for bringing me on. And, it was it's like the stars aligned because at ASM Microbe, they actually invited me to speak about rabies. So I had several talks there. They did a nice little TED style talk where I talked about bat rabies. So again, for your listeners, I hope you'll check some of those out and some of the other things that Mark will share. And I would be more than happy to join you again sometimes if you want to talk about some of the other areas I work in, which is antimicrobial resistance. Our medical lab advocacy, super important to get people into those majors. So just so appreciative of the work you're doing and the opportunity to speak with your listeners.
I wish you and your family the very best, and I hope to chat with you again soon.
Thank you so much.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with, as always, and in particular today, epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Rodney Rohde is chair and professor of the MLS program, College of Health Professions at Texas State University in San Marcos, Texas. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.